So thanks everyone. Thanks the organizer and the chairman. So it's my second time to this conference. I'm enjoying it. Nice to see old friends and meeting new friends. So I'm going to talk about some progress that we made in our lab. So this is somewhat an overview slide of what my lab is doing. So I'm mostly working with trying to understand RNA structure and the function. And we have developed many transcriptome-wide, transcript-specific-wide mapping technologies. And from then, we try to characterize them, knowing what protein it interacts with, and if they're functional RNA structural elements, so we develop tools to target it. And we'll be mostly focusing on talking about the targeting part in this aptamer conference. I think many of you here already know what G-quadruplex is. So I'll just go through it really briefly. So G sequence in DNA and RNA can fold, and then self-assemble into G-quartet, and stacking of the G-quartet further stabilized by the potassium ion will form this G-quadruplex. And from our mapping data, we've seen not only this G3N127 canonical G-quadruplex can be formed, but also other referred as structural subtypes of G-quadruplex, both in DNA and RNA. And also, sure, you've heard or read many papers on G-quadruplex and what they are involved in telomere, proto-oncogene, in the R loop and the RNA as well. And again, with many technology we've developed, we found these RNA G-quadruplex to be present in many different species. So indicating they're prevalent and probably they're doing something. And beyond the fascinating structure, I'm a trained chemist, so I always look at the structure. So there's a lot of biology involved already with DNA and RNA, and also there have been more and more evidence indicating they have association with different disease and metabolic disease as well. However, the current tools for targeting G4 are not very selective. So these are the four sort of major classes of tool currently: small molecule, PDS, NDC3, BRCA19, etc. Antibody, you can buy BG4, there's nanobody now as well as G4. Peptide, you can either from an endogenous protein or you can do it de novo and oligonucleotide approach as well. So they can, normally what they have shown is that it can be selective when you compare G4 versus non-G4, like duplex or single strand, but when you are trying to gain more selectivity, like RNA G4 versus DNA G4, RNA versus another RNA G4, it doesn't have enough selectivity to distinguish between the case. So, so this is, I think we start around 2020. So we were thinking about whether L-aptamer can be a potential solution to it. So just a few quick notes about this L oligonucleotide or L-aptamer. So the first is that naturally occurring RNA is in the B form. The synthetic form or the unnatural form is the L form. So they're mirror image of each other, as you can tell from the image here. I guess chemistry is nice. So if it's mirror image, then they basically have the same physical properties. So many of the assays that I'll show you, we actually do it on the B form, but it can directly translate to what happens in the L form. For example, UV melting and other assays. And because they're synthetic or unnatural, so they will be resistant to nucleic digestion. So this will be very good for cellular application or in vivo application later on when we develop the L-aptamer. There's been previous studies showcasing that. So the L, for example, L-aptamer will not interact with the tRNA target by the Watson-Crick base pairing. So the way to interact is normally through the Watson-Crick base pairing or tertiary interaction. So hopefully that can allow more specific recognition to be established. And L-aptamer is not something like I come up with. So in 1996, I remember it's Nature Biotech, there's two papers that they developed L-aptamer for small molecule and then later on with peptide, and then Joe Joyce, I think 2013 or 14 in the JAX paper, they have developed it for RNA capping. So the question I had when I started this project is whether we can use this technologies for G-quadruplex, and if so, then we can target, and because G4 are related to cancer, so maybe and neurological disease and viral pathogenesis, so we can have ways to slow them down or to have a way to detect them early on. So I mentioned this is my second time here. The first time I already present this the first generation of the platform RG4 Silex. So the protocol is out, so you can follow it step by step if you're interested in that. The issue with that first generation at the time is low throughput. We use standard sequencing, the old version of it. So they worked really hard. These are all PhD, Jamie is RA. So the success rate is very low. It takes them months to get the aptamer and to just for the identification process, not the characterization and application part. And then we developed second generation where we couple with NGS and also play a little bit more on template because now we start to understand what could potentially be a good aptamer that binds to G-quadruplex structure. Kelly is here, shows you the poster to talk about that. And then we also lowered the randomness a little bit in that generation. But still, it isn't where we want to be. It's improved. So throughput is better with the NGS and with the optimization of protocol. However, it still takes weeks. So then we have this third generation. So by combining human knowledge and past field experience by different PhD and research support staff in the lab. So Dan Yang, who was a PhD in my lab. So from many different aptamer that we have found, we see we can actually fix the stem region and just randomize the N15 region, and then we can come up with the relatively good aptamer. And I will talk a little bit more on this, using this as a representative example. So with that, we can become more high throughput. We can do it in weeks if you're lucky enough, of course. And also the success rate is quite high. So meaning that from the NGS data, it's not just the top one or the third one binds. So many of them, if you pick them, they will already show very good binding. And we're using this EBV as a case for this study. So EBV is a common virus that you find in human, and the adeno1 contains an RNA G-quadruplex that's important for the expression of the adeno1 protein, which is the key protein to maintain the EBV viral genome in a human. So to get into the detail a little bit on what we do with this platform, so this is the sort of the template and the library that we have. As mentioned, we can have a pre-structured library, and then we just randomize this region, and then you can go through the select cycle. There are a few additions to this platform too. We have this time, instead of using just the magnetic bead as a negative target, we actually introduce some other RG4 to further improve the selectivity process. And of course, the output, the readout is NGS, and we have the shorter randomized library. Long story short, 3 rounds of select. So Daniel already get a relatively good aptamer sequence here. And then we just showcase. So this is still the D aptamer L target. So try to find this and validate. So this is to the intended target adeno1 RG4 with 100 nanomolar KD. This is the negative target that we use, and the aptamer we pick so it doesn't bind very well to the negative control that we use. So it's indicating the select platform is working as intended. And then we flip, right? So it's mirror image. So if you have a D aptamer binding to the L target, theoretically your L aptamer should bind equally well to the D target. So this is what this shown in here. So now we use the L-aptamer B target. So you can see it's very similar to what I just showed you in this plot. And then we test this as the intended target, single-stranded RNA, hairpin stem loop RNA, hairpin stem loop DNA. They do not bind at all with the L-aptamer except for the intended target. We also show in here these are different DNA G4 we have in the lab, and it doesn't bind at all to the L-aptamer. So suggesting it is very good in terms of the selectivity. And then, so again, long story short, so we do some reported gene, and then we also do on the endogenous aptamer to see when we add the L-aptamer whether they can suppress the translation because the aptamer mRNA have repeats of G4. And then what we found is that, so by treating it with the L-aptamer, and then we have another L-aptamer that doesn't bind to the aptamer RG4 as a control, we found that the expression of the aptamer protein decreased, shown here, where the negative control doesn't have any effect. And these ones are the EBV positive NPC cell. So 90% of the case when people have nasopharyngeal cancer, they contain the EBV virus. So we use it as a case to suppress the protein shown here. So these 4 cell lines are all EBV positive cell lines. And we also look into the proliferation of the cancer cell as well. And again, we found that only with the case of our aptamer, the percentage of inhibition is shown in here. When we use the negative control, we do not have such case. So these 3 are EBV negative cell lines. So then you can see in this case, there's no aptamer, no EBV virus, and therefore no aptamer RNA. So this treatment will not affect their proliferation. So therefore, you will not see any effect when you have the aptamer or the negative control case. Okay, so that is the aptamer alone thing that we do in the lab. And then the next stage is so I also many people or many speakers and posters have shown you can couple aptamer with other stuff to either improve the performance, stability, or bivalent targeting, etc. So we're trying to do that with the L-aptamer as well. So one thing we do, I don't have time to go through the detail. So we have the L-aptamer conjugate with peptide, and the peptide we choose is a cell-penetrating peptide to sort of enhance the delivery without using lipo. And that's the study that we do in there, and we found the L-aptamer conjugate with the CTP cell-penetrating peptide can go inside the cell and do the target, the gene regulation sufficiently. We've also tried to understand more how to improve the performance. So L-aptamer binding to RNA G-quadruplex is non-covalent interaction, right? And it's just binding, right? So we try to, we have tried to incorporate DNA antisense oligo to it, so such that it can not only target the RG4, but also the flanking sequence of the RG4. So both to improve the binding affinity and selectivity. And what comes as somewhat a surprise to us at that time is this DNA-ASO can also trigger the RNA H mechanism. So that now we are not talking about just binding, we're talking about binding and catalytic cleavage. So in that case, it can strengthen the effect of the targeting and suppression of the translation. So this shown in that study. And then Kelly's work, here we'll have a poster. So basically, many of the orogenic aptamer, again, going back to the chemistry knowledge, so those ligands are achiral, meaning that maybe there's a possibility that we can use the L-aptamer, for example, spinach, right? Someone talked about spinach and pepper. So maybe we can convert that to the L form and conjugate with our L aptamer developed that targets G-quadruplex. So then this can become a G-quadruplex induced turn-on fluorescence. And Kelly have shown that we couple pepper with one of the L aptamer that targets G4, and we can showcase that, and we can do both in vitro and in cell imaging or detection and imaging of G-quadruplex. So still, there are many things that still needs to be done. Someone already asked me before in the poster section, or how do you get this L-RNA, how expensive it is. I think it's related to synthesis. So stability-wise, it's very stable. And then delivery is trying to improve it. Maybe some of you will ask me how they interact with G4 later on, so save that. And then we are trying to improve other aspects of it. And I think right now we are still more driven by the human knowledge, right? From the fair experience from different students, and we come up with a better template, better way to find a better aptamer. So we're also working with AI to see how we can improve it further. At the current stage of AI for RNA, it's very, very limited. So it's still a positive, like additive. It's more like a guide. But I hope in the future we can multiply it, so then maybe the AI can inspire us and tell us, okay, this is a better way to do it. This is a better aptamer than the one you start with. So hopefully we can achieve that stage, and maybe I'll present it again in the next 2 years or in the future. So take home message. So we've developed different platforms, and from those platforms we generate aptamer that can bind strongly and selectively. I use the adeno one as the demonstrated case in here. What it does is it will inhibit the adeno one translation. I didn't show the qPCR, so it doesn't affect the mRNA at all. So it's translational inhibition in here. So it has selective toxicity towards the EBV positive NPC cell or gastric cell. So potentially can be used for targeted therapy later on. So the poster from Kelly and Jenny. For acknowledgement. Thanks for the time and the attention.